Good morning, one and all. Myself, Mamaheshwari Kolipaka, working as Assistant Professor in AM Reddy Memorial College of Pharmacy, and today we are going to discuss about ELISA. Enzyme-linked immunosorbent assay. So today we are going to discuss about ELISA. First, coming to the definition, the enzyme-linked immunosorbent assay is a common laboratory technique which is used to measure the concentration of an analyte. Analyte usually antibodies or antigens in a solution. So enzyme-linked immunosorbent assay, which is very important laboratory technique or in vitro technique, which we are using to measure the concentration of either antibodies or antigens. So we are using antibodies or antigens as a analyte to detect the presence or to quantify, to measure the quantity of any analyte. We are using the technique that is ELISA, enzyme-linked immunosorbent assay. So why known as enzyme-linked immunosorbent assay? Antigen or antibody of interest is absorbed onto plastic surface that is sorbent, and antigen is recognized by specific antibody that is immuno. This antibody is recognized by second antibody that is which has enzyme attached, enzyme linked. Substrate reacts with enzyme to produce product, usually colored product. So enzyme linked immunosorbent assay. How the name given means antigen and antibody of interest is absorbed to a solid surface that is plastic surface, which we can call it as sorbent, and antigen is recognized by a specific antibody that is with the help of immunological reaction, immunological response. Basically, antigen will react with the antibody or antibody will react with the antigen. This is said to be immunological response. And how we can come to know whether the immunological reaction has been taking place or not. That is, we are coming to know with the help of the enzyme which we are linking to either antibody or antigen which is reacting to the opposite one. So final product will be produced in a manner that we can easily identify with the help of its different color. So colored product will be produced. So based on this process, we are given the name enzyme-linked immunosorbent assay. Next, coming to the history of ELISA. Radioimmunoassay was first described in a scientific paper by Rosalind Sussman Allo and Solomon Berson, published in 1960. In 1971, Peter Fernan and Eva Evan Engvall as at Stockholm University in Sweden and Anton Schurz and Bachman women in the Netherlands independently published papers that synthesized this knowledge into methods to perform the ELISA or enzyme immunoassay. So these are the scientists who have published the technique of ELISA. So first coming to the basic terms, solid phase, usually a microtiter plate or microtiter plate well that is having 18 to 12 well format. So if you are considering these are the 8 wells and these are 12 wells, so 18 to 12, total 96 wells we have in this microtiter plate. Basic terms, adsorption, the process of adding an antigen or antibody diluted in buffer so it attaches to the solid phase on incubation. Adsorption, so it is nothing but attaching or annealing the antigen or antibody in a diluted buffer so that it is getting attached to the solid phase or solid plate. Washing, the simple flooding and emptying of well with a buffer solution to separate bound from unbound reagents in ELISA. Antigen, Any molecule that elicits the production of antibodies when introduced into the body. So antibodies, proteins produced in response to the antigenic stimuli. So we are all very familiar with these words, antigen and antibody. Antigen is foreign particle which is coming from the outside to our body, into our body. And antibody, in response to the presence of antigen, antibody is a protein which is produced, synthesized by our immune system to defend, to fight against to the antigen. Enzyme conjugate means an enzyme that is attached irreversibly to an antibody. Example, horseradish peroxidase. Chromosome, a chemical alters color as a result of an enzyme interaction with substrate. That is a color reaction used as signal. Example, trimethyl benzidine. So whenever we are using this ELISA technique, we are linking the antibody or antigen with the help of enzyme. So whenever we are adding the substrate, in the presence of that enzyme, the substrate is getting converted to a product. Most of the times, this product will be in colored format, so that we can come to know the reaction really it is taking place or not. So the colored product we are using, that are nothing but chromogens. And stopping the process of stopping the reaction of an enzyme on substrate, on a substrate. Reading spectrophotometric measurement of color developed in ELISA. So we can come to know about the quantity, how much amount of analyte is there, that is according to the color intensity produced in the form of product. So based on the product color intensity, by measuring the intensity of the color of the product with the help of spectrophotometry, we can come to know about the quantity of that analyte which is present in that mixture or sample. Principle of ELISA. Based on basic immunological response, lock and key concept, antigen antibody. One is key and one is lock, acts as a lock. So key fits exactly into the lock. And enzyme conjugate substrates bound to a secondary antibody that binds with the antibody antigen complex. Equipments, micro well plates or micro titer plates, flat bottom polystyrene plate contains 18 to 12 wells holding 350 microliters each. So what is the capacity of each and every well? If you ask, answer is that 350 microliters. And equipments, multi-pipette or micro pipettes. These are said to be micro pipettes because microliters capacity we will deliver into the micro titer wells. An 8-channel 100 microliter pipette is a good help for even small scale work. Equipments, washing device. So we need to have washing device to wash out or to remove the unbound antigen or antibody. Then only we can come to know the exact presence, whether they have attached already adsorbed one already to the existed antibody or antigen in our sample. If any analyte is there, that will get attached exactly opposite to one, so that we can come to know. So if any unbound particles are there, we need to wash out them. So for that, we need to have washing device. Manually operated washing devices may be of use, particularly when there is a risk that the samples tested in ELISA contain infectious material. So must be collected for subsequent disinfection. Equipments, microplate washer. These are very efficient with usually low carryover contamination. Low carryover contamination. So reagents used, coating buffer, that is 0.01 molar phosphate buffer plus 0.15 molar sodium chloride. Diluting or washing buffer, 0.01 molar phosphate buffer and 0.5 molar sodium chloride, as well as 0.1% Twin 20. Blocking buffer, bovine serum albumin, that is acronymed as BSA, and enzyme horseradish peroxidase, HRPO. Chromogenic substrate trimethyl benzidine, and stop solution 0.5 molar sulfuric acid. General procedure. What is the basic procedure involved in the ELISA? So first of all, we need to take antigen, that is a sample is added to the plate. Blocking buffer. Blocking buffer is added to block remaining protein binding sites. Next, a substrate primary antibody is added. A suitable secondary antibody, HRPO conjugate, HRPO conjugate is then added, which recognizes and binds to the primary antibody. TMB substrate is added and is converted by HRPO to detect the form. So this is the basic procedure. First of all, we have to add antigen to the microtiter plate. Then we are adding the primary antibody. This is our primary antibody. This is our antigen. So primary antibody, then we are adding the secondary antibody, which is we are linked with the help of the horseradish peroxidase enzyme. So whenever we are adding the substrate, that is trimethyl benzidine colored product, substrate colored substrate, we are adding. That is basically it is initially don't have color. Upon it is converting to the product, it will change its color. It will look like a different colored product. So this is the basic concept here. The basic principle is if our antigen is exactly against this antibody. So that is this primary antibody exactly against this antigen. Definitely it will go and bind to the primary antibody will get bind to the antigen. Then we are adding the secondary antibody, which is actually linked with the HRPO, human radish peroxidase enzyme. Then it is getting converted to a color product upon the addition of the substrate. So next, coat the plate with the capture antibody. Then, first of all, we are coating the plate with the antibodies. Then, um, second step is blocking plates. If we are adding additional antibodies, also there is no space, so that there is no binding of that antibodies because already antibody existed. Next, add sample. Now we are adding the sample. Next, add analyte, that is linked antibody, secondary antibody, enzyme linked antibody we are adding. Then substrate we are adding. Then the product will be produced that is appeared in the different color. Types of ELISA on the basis of detection, colorimetric ELISA, assay to determine the antibody concentration. So first of all, immobilized antigen. We are making the antigen immobilize by annealing this antigen to the solid surface, that is plastic microtiter plates or polystyrene microtiter plates. Next, we are adding the primary antibody. This is our primary antibody. Then we are adding the secondary antibody, actually, which is linked with the enzyme. Then we are adding the substrate so that this substrate is getting converted to the product with the help of the enzyme, which is here in green color. With the help of this enzyme catalytic activity, the substrate will getting converted to the product. This is colorimetric ELISA. Colorimetric means color produced product. Product will be appeared in a different color. So based on the color, we can come to know whether they are whether the sample is having the exactly suitable antibody, exactly antibody which can able to bind to our antigen. Next types of ELISA, chemiluminescent ELISA, assay for the quantification of an antigen in a biological sample. So here we are using the antibody that is sticker to the polystyrene plate. Then we are adding the sample, which is in which we are expecting the presence of antigen. Then this antigen will bind to this antibody. Then labeled antibody we are adding. This is actually secondary antibody because primary antibody initially we have taken into the plates, then antigen binded, that is antigen present in the sample. Now we are adding the secondary antibody, which is linked with the enzyme. So basically this secondary antibody is also bind to the antigen. Then we are adding the substrate. This is chemiluminescent ELISA. So we can plot a graph by taking analyte concentration on y-axis, sorry, x-axis, and signal strength on y-axis. So signal strength is directly proportional to the concentration of analyte. So this is chemiluminescent ELISA. Next, coming to the competitive fluorescence ELISA. So here, in this case also, we are taking the antibody onto the microtiter plate. Then we are adding addition of antigen and antigen conjugated enzyme complex, binding competition between the antigen and antigen conjugate. So single antigen and antigen conjugate, that is enzyme linked antigen and normal antigen we are taking. So whichever is more in concentration, that will get bind to the already existed antibody. Then we are detecting the signal to detect the quantity, which one is more. So here also we can plot a graph in between the analyte concentration and the signal strength. So signal strength is obviously inversely proportional to the concentration of analyte. So this is the competitive fluorescence ELISA. Next, coming to the basic types, non-competitive, competitive, and multiple and portable. So non-competitive ELISA is again differentiated into three types: direct ELISA, indirect ELISA, and sandwich ELISA. Already we have seen competitive. Next, coming to the non-competitive. Direct ELISA, it uses a primary labeled antibody that react directly with the antigen. And it can be performed with the antigen that is directly immobilized on assay plate. Now, not widely used but common for immunohistochemical staining of cells and tissues. And indirect ELISA. It utilizes a primary unlabeled antibody in conjugation with a labeled secondary antibody. Secondary antibody has specificity for primary antibody. So direct and indirect ELISA, here we can observe. This is direct ELISA. First of all, this is the plastic or cell surface, that is polystyrene surface, to which we are annealing or attaching. The antigen is absorbed onto this surface. Then we are adding the sample, which is having antibodies that is tagged with an enzyme. So that whenever we are adding the substrate with the help of this enzyme, substrate is getting converted to a product. So this is direct ELISA. Directly, there is no secondary antibody or secondary antigen. Directly, we are tagging the antibody with the enzyme. So this is direct technique. Next, coming to the indirect. In indirect, antigens are getting adsorbed to the surface, solid surface. Then we are adding primary antibody. Again, we are adding secondary antibody. Primary antibody, actually our sample. Secondary antibody, which is tagged or linked with the enzyme, so that this secondary antibody actually linked with the uh, primary antibody in this region, in this region, that is um, basically antibodies are having two regions, FC region and which is linked, this region only linked with the antigens. This region can bind to the other antibody. So the secondary antibody will come and bind to the primary antibody in this way. So whenever we are adding the substrate is getting converted to a product. This is indirectly we are detecting the presence of sample, the presence of primary antibody. That is why this is said to be indirect. Non-competitive ELISA, sandwich ELISA. Antigens like tumor markers, hormones, serum proteins may be determined by using this sandwich ELISA, and antigens in the sample bind with the capture antibody and become immobilized. The antibody of the enzyme conjugate bind with the immobilized antigen to form a sandwich of antibody, antigen, antibody, and enzyme bound to microbe. So this is sandwich. Actually, one antibodies are we are absorbing, making to absorb onto the surface. Then we are adding the sample. So if sample is having the antigen, definitely this antigen will bind to this antibody. Again, we are adding the secondary antibody that is linked with the horse radish peroxidase. So this is also linked up with the antigen. Now, if we are looking at this picture, this is look like a sandwich. In between the two types of antibodies, our antigen will be placed. Our antigen will be existed. So this is said to be sandwich ELISA. So whenever we are adding the substrate, it is getting converted to the product that will be produce some sort of light, some amount of light that is we can detect with the help of chemiluminescence detectors, or sometimes the product will be a colored product. So by looking at the color change, we can come to know. So this is our indirect ELISA. Then we are going to discuss about sandwich ELISA, in which we can observe the presence of antigen in between the primary antibody and secondary antibody. Here also we can observe the color, the product, colored product. Then this is competitive ELISA, depending on the concentration. Depending on the based on the concentration, the thing will be bind to the existed one, that is either antibody, um, primary antibody, secondary antibody, both we have added. Primary antibody actually present in the sample and secondary antibody we are adding. So according to the concentration, either of one will be added to the antigen, so that upon the addition of the substrate, we can come to know about the colored product formation. Next, coming to the multiple and portable ELISA, a newer technique uses an solid phase made up of an immunosorbent polystyrene rod with 8 to 12 protruding gives. The entire device is immersed in a test tube containing the collected sample, and the following steps: washing, incubation, and conjugate and incubation in chromogenes are carried out by dipping the ogives in microwells of standard microplates pre-filled with reagents. So these are the pins and rags. Modified ELISA enzyme interface with antigen antibody interaction. Secondary antibody is often labeled with a very small molecular substance, that is biotin, and a specific binding protein for a biotin. Evidin is conjugated with the enzyme such as HRP. So modified ELISA is nothing but here we have already coated the antibodies, that is um, monoclonal antibodies coated on microwells. Then target protein we have given the sample. So it is binding to the already existed antibody. Then we are adding the other antibody which is labeled or linked with the biotin. So that this is also binding to the antigen. Then we are adding the streptavidin as a substrate, so that it is getting converted to a colored product. This is modified ELISA. Reading: measure the absorbance at 450 nanometers with the help of ELISA reader. Calculate the absorbance for each sample and reference. Ascent software for the calculation of results can be used. So results that may be positive, negative, or assay control. So different samples we can take, and we can detect the presence of antigen or antibody, so that we can come to know the diagnosis of the disease. Next, troubleshooting in ELISA. If the negative um, results are giving negative controls are giving positive results. Contamination of the substrate, resolution, enzyme labeled antibody control themselves. Inadequate rising of inadequate rinsing of plates, inadequate blocking of plates. These are leading to the negative controls. Next, if very little color has developed for positive controls and the best and the test samples, Now check the dilution of the enzyme labeled antibody, the concentration of the substrate, wash buffer not adequately drained after every wash step, inadequate incubation times, enzyme conjugate defective or inhibited by contaminant substrate defective or contaminated microbial plates poorly coated. Color has developed for the test samples but not the positive control. Check the source of positive control. Then expiry date and storage. So we have to do all these things in such a way to prevent these errors in the results. The color can be seen, but the absorbance is not high as expected. Check the wavelength. So use of exchange type pipettes. We have to use um, hygienic sterile pipettes to prevent the errors. And we have to wash the plates in such a way there is no sticky things on the plates. Reagents, we have to use the reagents that is quality reagents, and we don't use expired one. Don't use the same reservoir for multiple reagents. So we know we should not store the different reagents in the same container. Don't return the reagents to the stock. So plate cover. During the incubation, well plates should be covered using the plate cover. Plate cover is effective only under suitable conditions, that is room temperature and humidity more than 50% and air stream less than 0.2 meters per second. Precautions: coating of wells. Coating of wells should be proper with the addition of blocking solution. Improper coating gives us false results. Advantages of ELISA. Reagents are relatively cheap and they have long shelf life. It is highly specific and sensitive. No radiation hazards occur during labeling or disposal of waste. Easy to perform and quick procedure. Equipment is widely available. It can be used to variety of infections. It can be used on most type of biological samples like plasma, serum, urine, and cell extract. Disadvantages of ELISA. Measurement of enzyme activity can be more complex than the measurement of activity of some type of radioisotopes. Enzyme activity may be affected by plasma constitutes. Kits are not cheap. They are specific to particular antigen but won't recognize other antigens. False positive or negative possible, especially with the mutated or altered antigens. Limitations are also there. Results may not be absolute. Antibody must be available. Poor producer or interference. Concentration may be unclear. False positive possible. Antibody already present, false negative possible. Applications. Number of applications are there. We can detect the antigen or antibody, that is literally we can diagnose the disease. Screening donated blood for evidence of viral contamination by HIV-1, HIV-2, hepatitis C, hepatitis B. Measuring hormone levels, we can measure the hormones. HCG, human chorionic gonadotropin, that is pregnancy test kit, is based on the principle of ELISA. And luteinizing hormone, yeah, we can detect with the help of ELISA. TSH, thyroid stimulating hormone, T3, T4. We can detect with the help of ELISA. Detecting infection, sexually transmitted diseases like HIV, syphilis, chlamydia, gonorrhea. We can detect with the help of ELISA. And hepatitis B, C, toxoplasma gonadie. Detecting illicit drugs, illicit trafficking, we can do with the help of ELISA. Detecting allergens in food and house dust. So these are the references we can use to get more information about the ELISA. Thank you.